So it's wonderful to be able to talk to you today and showcase some of the MND research we're conducting at the Florey Institute. And I'd like to thank you all for attending up front and for those of you who have travelled long distances. So the overview of today's lecture is I'll give you a history of MND. So what are the origins of MND? I'll talk about the causes of MND. So what do we understand in terms of genes and environmental contribution to motor neuron disease. I'll give you a quick update on clinical trials for MND, and then, like I mentioned, I'll showcase some of the exciting research we're producing at the Florey Institute. So if I start with the history of MND, this is quite an old disease, and its origins actually date back about 200 years ago. And there was a chap called Charles Bell, and he was a neurologist, theologian, neurosurgeon, clerk, amongst other talents that he had. And he's been attributed to be the first person to describe motor neuron disease, dating back to 1824. And it was in this publication in which he had a woman, and her name was Mrs. Berkeley Square, and he described her in this way. So he said that she had totally lost the use of all the voluntary muscles. Her legs and arms lay motionless. Her tongue refused utterance. However, her eyes moved and were expressive. So this is the very hallmark of motor neuron disease, is that these patients lose voluntary muscle movement, but their eye movements are intact. So about 50 or so years later, the next event was in Paris. So there's a hospital called the Salpêtrière Hospital, which is world famous. This used to be an asylum. And there was a neurologist called Jean-Martin Charcot, and he revolutionised 20th century neurology because he turned this once asylum into a world-class hospital for neurological disorders. And this is a picture of him. That's actually a photo of me trying to follow in Charcot's footsteps last year. So I took a trip to the Salpêtrière Hospital. And what he did was he took Bell's observations further, one more step, and that is he had 20 cases of motor neuron disease, and he autopsied 5 of them, and he looked down the microscope, and what you're looking at here is a cross-section of spinal cord from someone affected with motor neuron disease. And what he noticed was there was a loss of nerve cells in this region, which we call the lower motor neurons, and this produces muscle wasting. And he also noticed the loss of nerve cells in this region of the spinal cord, which are called the upper motor neurons. And the loss of these cells is responsible for muscle twitching and spasm. So his genius was to actually connect the dots. So he was able to relate the symptoms to the underlying pathology. And he gave a description that some patients showed both progressive muscular atrophy and spastic contractures. So that was Charcot's genius, to link the pathology to the clinical signs. Then the story continued. So about 10 years later, now we move to North America. There's a state in the corner called Vermont, which is the Green Mountain State. And there was a family, quite a large family, known as the Farr family. And Erastus Farr II, he was a farmer, he was a labourer, he was out in the field, and one day he noticed he had weakness in his left leg, and he developed a limp, and this became a problem. So he thought he would seek some medical advice, and he undertook a journey. No one's quite sure how he did this, but he managed to cross the border into Canada, and he had an appointment with a neurologist in Montreal, which is quite extraordinary. So he took the first step to identifying what his disease was. And he had an appointment with a neurologist called Dr. William Osler. So Osler's quite famous in medicine because he founded Johns Hopkins University and School of Medicine. And Dr. Osler made the following observation. So he said that the left leg appears to be smaller than the right, owing to uniform wasting of muscles, and twitchings have also become general and occur irregularly in different muscles. So what Osler was describing was similar to Charcot and Bell. So they've all come across the same disease. But Osler took it one step further. He started to talk to Erastus II, and he asked him about his family. And Erastus said that he wasn't the first to have this disease. In fact, 13 members of his family were affected. So this is a family tree of the family. This is Erastus here. And you can see here that he has siblings which are affected by MND, and also nieces and nephews, and his parent and grandparent. And what Osler found was, he said that there's a clear hereditary component to motor neuron disease in this family. And what we do know is that this family, the Farr family, have been ravaged by motor neuron disease for over 150 years, and they're still living with this disease to this day. So this is a picture of family members throughout time who have succumbed to motor neuron disease. And this is just one family in the world affected by this disease, and we still don't have a cure. So I think it was quite extraordinary that Erastus Farr took the steps to visit Osler, and Osler was able to identify a hereditary component. So nothing much happened in the MND space until about 1930s, in which a very prominent baseballer by the name of Lou Gehrig. So Lou Gehrig he was known as the Iron Horse. He was a champion baseballer. He played 17 seasons straight. And then one day he noticed that he was developing fatigue and weakness on the field. He wasn't able to slug the ball, and he actually was diagnosed with motor neuron disease. And he had to retire, sadly, from his professional career, and he gave a very emotive speech in which he considered himself the luckiest man on the face of the planet, despite the bad odds that he'd been dealt with. And this is a photo here of him retiring from his professional career. And he actually died about 2 years after this speech. So Lou Gehrig's disease is synonymous with motor neuron disease in the United States of America as a result. So he became the national emblem or icon for MND in America. So again, nothing happened for a good 70 years until a few years ago there was this phenomenon known as the Ice Bucket Challenge, which I'm sure many of you are familiar with, in which it was a viral campaign in which various celebrities and members of the public were able to drench themselves with ice buckets and donate funds to motor neuron disease research. And this is a picture of Bill Gates. So he invented his own elaborate contraption. We have Mark Zuckerberg, founder of Facebook, and even Oprah Winfrey got involved. And this is actually Jeff Donnan, our director of the Florey Institute. You've spoken to this audience before, and he got challenged by another director from a neuroscience institute in Australia. So Jeff was challenged, and he did formidably with the task. Now, President Obama was asked to undergo the Ice Bucket Challenge, so he politely declined. However, I can tell you his body double did perform the challenge, as shown here. And this was a phenomenal success because it raised $3 million in Australia and collectively worldwide over $200 million for MND research. And then the success continued. So at the Oscars in that particular year, this actor, Eddie Redmayne, he portrayed Stephen Hawking. So Stephen Hawking is a very famous example of someone who's living with motor neuron disease. Although he has an extraordinary course of motor neuron disease, he's been living with it for 50 years. And it was actually this actor's portrayal of Stephen Hawking in this movie called The Theory of Everything that earned him the Best Actor Oscar. So this brought some recognition and notoriety to the disease. And then we had a homegrown hero come forward, and that was Neil Danaher. So many of you will be aware of Neil Danaher. He came out publicly with his diagnosis a few years ago, and he's led a very high-profile fundraising awareness campaign. The big freeze at the G, so the third one is next week on Queen's birthday, and he's a very strong ally of the Florey Institute, and he's able to bring the Premier Victoria into the laboratory, and we have a very close contact with him, and he's certainly been helping us with funds. So let's get to the facts about motor neuron disease, and these are the facts, and that is that in Australia we know that 2 people are diagnosed every day with motor neuron disease, and this disease is unlike other disorders of the brain such as Alzheimer's and Parkinson's because it's rapidly progressive. So it's thought that patients have an average survival of 27 months from diagnosis. Sadly, we have no effective treatments or cures for motor neuron disease, and as a result we're losing about 2 Australians a day from this disorder. Now, MND can affect anyone, and what we do know is, even in the context of this lecture theatre, we know that 1 in 300 people have a lifetime risk of getting this disease, which is not trivial. What's disturbing is that the incidence of motor neuron disease is rising. So if we look at the last decade in Australia, the incidence or the number of deaths from motor neuron disease have doubled over the last 10 years. So this is a disease which is on the rise, and we know that 1 in 200 deaths in Australia were related to motor neuron disease. So this is a serious growing problem. What do we know about the costs of MND to society and the economy? So this is a very expensive disease to manage. And this is shown here in a recent economic analysis performed by Deloitte, in which they calculated that motor neuron disease cost $2.4 billion in 2015. And I imagine this has increased since. And this is due to the large burden of care associated with the disease. This is a challenging disease to manage because it requires multidisciplinary care from neurologists, speech pathologists, nutritionists, respiratory physicians, and so on. So it's quite a complicated disease to manage, and it's an expensive problem. So that brings me to the next part of my talk, and what do we know about the causes of motor neuron disease? So what we do know is that in the case of the Farr family, that they had a clear hereditary form. And we know that 10% of cases of MND are inherited, and that's shown here in this pie chart, whereas 90% of cases are what we call sporadic. And sporadic simply means out of the blue, no family history. And this is the most common form. So Neil Danaher has sporadic motor neuron disease. So his brothers, who also played AFL, are not affected, and he has no family history. This is another way we can show this. So what we mean by inherited disease is that this individual here is affected and it's passed on the gene to his daughter and son, and they've passed it on to their children. And this is an example of what we call dominant inheritance. So the gene is passed on and to every generation of the family. Sporadic MND, so as I mentioned, there's no positive family history in sporadic MND. So here we have about 10 individuals who are not related through bloodline, but 3 of them have developed motor neuron disease. So the question then is why? And that brings me to this slide. So what is the cause of sporadic MND? Well, I could give you 100 reasons or theories as to some of the ideas about what causes MND. And what I thought I'd do today is just outline a few of these which are most popular. So what we do know is that cigarette smoking increases the risk of motor neuron disease by 2 times. So there's a positive association with cigarette smoking. And now this is actually thought to impair lung function, so it makes sense that would increase your susceptibility to motor neuron disease. This is generally bad for your health anyway. Professional sports, so there's some good evidence now that if you think of Lou Gehrig and Neil Danaher, that certain professions involved in athletic sports and elite athleticism, there is an association with motor neuron disease. Military service, so a lot of Gulf War veterans, they serve and return to the United States, and then they have an increased risk of developing motor neuron disease. And the Department of Defence is very interested in looking at this. This is actually a picture of a virus. This is a microscopic virus particle. So there's an idea now that there could be a virus which lurks within the brain, and it becomes reactivated in motor neuron disease and can cause damage. And then lastly, this is another microscopic organism, which is blue-green algae, which I'll talk about. So if we start with professional sports and athleticism, so the most famous example of a potential association here is Lou Gehrig. His career was cut short. And there was a paper published in a medical journal about 10 years ago in which they had tracked a club, a soccer club in Italy, and they determined that these players had a 6 times increased risk of developing motor neuron disease over a decade compared to anywhere else in the world. And this is interesting for several reasons, because there are several potential explanations. So is it the extreme physical activity and training involved? Is it due to repeated microtrauma to the brain? Could it be resulting from potential abuse of steroids and performance-enhancing drugs? Could it be exposure to the environment? So if you think about they playing on fields, there's this potential exposure to fertilisers and pesticides. So this raises a couple of different ideas about what could be responsible for this association. Now let's move on to viruses. So when we talk about viruses in MND, we're talking about a specific type of virus called a retrovirus. So what is a retrovirus? They are cool looking. So there was a paper published about 2 years ago which showed that we're looking here at spinal cord sections from patients with MND, and these are the motor neurons here, and they were loaded with viral particles compared to the healthy control. So this was quite unusual, and it's introduced this theory that perhaps in MND that there are viral genes. So when we think about all our genes, about 10% of our collective genes are actually viral. And what happens throughout evolution is that viruses have inserted themselves into our genes, and it's thought that for some reason in motor neuron disease, these viral genes are switched on and they produce live virus which can damage motor neurons. And this is certainly consistent when you think about the clinical pattern of motor neuron disease, in that it spreads insidiously. So it begins in one finger and then will invade the whole hand and move to the other hand. So this would be consistent with an agent that's able to spread throughout the brain. So the idea here is that the viral genes are turned on in motor neuron disease. They produce viral particles which assemble live virus which can then damage motor neurons, infect neighbouring motor neurons. And there's currently a clinical trial at the moment that will test this idea, and it's using an agent called Triumec. So Triumec is an antiviral agent. It's used for patients with HIV, and this drug has been repurposed for exploration in motor neuron disease. And there's a clinical trial in Australia which is testing the ability of this drug to perhaps slow motor neuron disease in these patients. So now I'm going to move on to blue-green algae. So this is this quite harmless seemingly looking organism. And in order to give you some background to this, I wanted to tell you a story. And this is a story that evolved back in the 1950s in an island called Guam. So Guam is in the Pacific. It's this little island up here. And what was interesting was the incidence of motor neuron disease on this island back in the 1950s was 100 times greater than anywhere else in the world. So this was quite puzzling to medical researchers. And on this island, there was a tribe of people called the Chamorros. So they are the native people of Guam. And they were afflicted by a disease which they called litigo-bodig. And this translates as paralysis and tremor. So here we have an affected individual. This man's poor wife has clear signs of muscle wasting and paralysis similar to motor neuron disease. So this was quite puzzling. And it got medical researchers thinking about, is there some potential agent in the environment which could be contributing? And they began to suspect that perhaps it was a plant. And this is a plant known as a cycad. And this produces these seeds, and these seeds are edible. And what was interesting was the Chamorro people knew that this plant was toxic. It contained a toxin. And what they would do is they would harvest the seeds and soak them and wash them repeatedly to rinse out the toxin. And then they would grind the seeds into flour and make their tortillas. So this got the medical researchers quite puzzled. They thought if they knew about the toxin and they were removing it, how was this getting into the food chain? And then the answer came from the observation that fruit bats, which were native to Guam, were feasting on these seeds from this cycad plant. And then it was uncovered that the locals, so the Chamorros, were quite fond of fruit bats. So here we have some examples of culinary delights using fruit bats. So they had them for breakfast and lunch and dinner, and they were crazy about eating fruit bats. And so this theory evolved that perhaps in the roots of this cycad plant, it was found that there was blue-green algae, which makes a toxin. And this toxin, if we go over here, concentrates into the seeds. The seeds are devoured by the fruit bats, and then the fruit bats are consumed by the local Chamorro people. And this was actually thought to be the mode of transmission. This is how the toxin was getting into the food chain. Once this was theorised and discovered, there was a ban put on the consumption of flying foxes in Guam, and the incidence of motor neuron disease is virtually nil. So this is some good evidence that blue-green algae could be causally involved in motor neuron disease. Now, how is any of this relevant? I doubt many of you eat fruit bats for breakfast. Well, it is relevant because it brings us back to algal blooms. Algal blooms are quite prevalent in our waterways and reservoirs, and this is an example of, we're looking here at the Murray River about a year ago, and so parts of northern Victoria and the Riverina are plagued with algal blooms. And obviously, in the short term, this is not safe to swim in or drink because it will cause a nasty lung function. But now there's this idea that the incidence of MND is actually quite higher in regions associated with algal blooms in the Murray River, and perhaps that this is a potential source of a toxin that could cause motor neuron disease. So this is quite an interesting idea. So let's come back to inherited motor neuron disease. What do we know about the genes that are responsible for inherited MND? And what we can do as geneticists and molecular scientists is that we're able to read the DNA sequence, so the coding sequence, the genetic code within patients. And the way we do this is we would take an individual here who's not affected by MND and read their genetic code, and that's shown up here. And then over here we look at the affected individual, and what we can see here is there's a mistake in the genetic code, and this is causing the motor neuron disease. So what I wanted to tell you was it was quite striking that if we go back and look at the timeline here, so William Osler described this inherited motor neuron disease in about 1880, and what's extraordinary is it took over 100 years to identify the gene responsible for motor neuron disease in this family. And that gene was discovered back in 1993 by an international group involving Australian scientists, and that gene was called SOD1. So what I've got here is a graph, and this is showing you the evolution of progress in motor neuron disease genetics. So we know that this gene SOD1 was discovered back in 93, and it's responsible for the Farr family's disease. What's really encouraging from this graph is that we've had a very steady exponential increase in discovering different genes causing motor neuron disease, and we're at a point now where we've gone from 1 gene to 20 genes, and particularly in the last 5 years. And another way we can graph this is in a pie chart. So if I took you back 10 years ago, I would tell you that we only knew of 1 gene responsible for MND called SOD1, which accounted for 20%, and we didn't know what the other 80% was. However, in current age, what we do know is we've solved about 70% of this puzzle, and we're actually chipping away at this last 30%. And it's thought that in the next 5 years, this will be 100% solved. So this will give a genetic diagnosis to any family with motor neuron disease. This will give confirmation. So this is quite steady progress. So what we have here is a cluster of, these are chromosomes which carry all your genes. We have an increasing number of genes responsible for MND. And what these genes do is they point to the problem. And this is the problem that we have, is we have many genes pointing towards what we call biological complexity in motor neuron disease. So this is a nice cartoon of a motor neuron. And what you can see here is this motor neuron has several events within it. So it has several problems that are contributing, or may be contributing to disease occurring along the length of it. And then to add to this complexity, you've got some external cells which are interacting with the motor neuron. And collectively, this causes a cataclysm of destruction within the motor neuron, which brings about its death. So now I'm going to talk to you about the clinical trials for motor neuron disease. So what do we know in terms of developments in this area? And this is quite a busy figure, but I think it's quite useful. So what we'll do is we'll just focus here on the right side of the graph. What we're doing here is we're comparing 2 diseases. So the first one is MND or ALS, as it's known interchangeably, and the other is a disease called frontotemporal dementia. So this is a type of non-Alzheimer's dementia. So the first thing you'll appreciate from this graph is that these are all drug candidates. There's a lot going on the right side, which is the MND. So there's more progress in MND. And we can think of this particular graph as almost like a dartboard. And what we're trying to do is these are different phases of clinical trials. So 1, 2, 3, 4, and we're trying to end up in phase 4. So phase 4 is marketing of drugs. And what we can see is lots of different drug candidates are on this board, and they're all trying to move into the centre. And I'll just point out 3 things about this graph. So the first is that about a month ago, there was a drug which is being clinically approved for motor neuron disease called edaravone in the United States and also Japan. So this is a huge breakthrough because we haven't had a clinically approved drug for MND for over 22 years. However, there are some conditions regarding this drug, so it's actually very difficult to administer. It requires a patient to visit a hospital. They have to be an outpatient. It's given intravenously. It's also hideously expensive, so it's $150,000 a year. So there's some issues that need to be dealt with before the Australian equivalent of the FDA can get this drug approved. The other thing I'll point out is that there are currently 2 drugs under clinical trial, and these are conducted in Australia. So we have antiviral drugs. So I mentioned before about that Triumeq drug. So this is actually in phase 2 trial in MND patients in Australia. And lastly, a drug which came out of Melbourne University and the Florey Institute called a copper drug, copper ATSM. That's currently undergoing phase 1 trial. So this is incredibly exciting. We've never had a situation where we've got 2 clinical trials in Australia concurrently. So what I will tell you is that clinical trials are conducted across 4 sites in Australia, and we're fortunate to have Neuroscience Trials Australia embedded within the Florey Institute, and they're actually involved in coordinating these drug trials. And our trials are conducted at the Bethlehem Hospital in Caulfield South, but we also have other sites in Sydney and also Brisbane. So Australia is very active on the clinical trial front for motor neurone disease. Okay, so this brings me to the 4th part of my talk, which is I'm going to show you some research coming out of the Florey for motor neurone disease. So I'll just give you an overview of the Florey for those of you who don't know. So we are the largest brain research centre in the Southern Hemisphere, and we have over 500 scientists working on different areas of the brain, and with 150 students, PhD students. And we've got 2 campuses. So this is what we call our basic science campus. So here we're involved in discovery using model systems. And then we have a clinical campus at the Austin Hospital, which would coordinate clinical trials. The Florey it has an emphasis on what we call from bench to bedside translational medicine. So that's taking drugs discovered in the lab to the patients in the quickest time frame possible. And this is largely facilitated by Neuroscience Trials Australia, which are coordinating both the antiviral and copper ATSM trials in MND. We also have very advanced brain imaging platforms, and I remember about a year ago there was one installed. It was a 7 Tesla MRI, and they had to use a crane, and it was quite a commotion outside. So the Florey has a strong disease focus. So MND is one of the areas that we research, but we're also interested in other debilitating disorders that affect the brain and the mind, such as Alzheimer's, Parkinson's, and MS. And it's actually thought that by using this structure within the Florey that we can collaborate across different groups, and there may in fact be something in common between Parkinson's and motor neuron disease and vice versa. I just wanted to show you the lab members. Many of them are in the audience today. So this is the structure of the lab. We're quite a large group. We're growing. We're always expanding. And we are supported by research assistants in the laboratory. We have our own GP and clinician in the group. And then we have our army of postdocs. So these are scientists who hold PhDs and they're researching MND full-time. And then we have PhD students, and they're really the lifeblood of the group, as well as honours students. So we have quite a large team working on MND. And we can't do this alone, so we collaborate across interstate. So we have collaborations in New South Wales, Queensland, South Australia and Tasmania. So we're actively collaborating with other MND groups. The collaboration has to occur on the international front. So here's an example of different institutions we're working with. And I just wanted to point out that if you think about 200 years ago, that the epicentre of MND research was at the Salpêtrière and McGill University, then these are 2 of our collaborators in modern times. So we're very well placed in terms of collaborations. And this is our approach to motor neuron disease research. And that is we have this pool of genes that are responsible for MND, and we're able to model these genes in the Petri dish. So here we have, this is a motor neuron that we've grown, and it's healthy, and this one's affected by an MND gene. You can see clear signs of stress and sickness to this cell. So this is a drug screening tool that we can use. And then what we can do is advance our studies to animal research. So animals are vital to MND research or medical research in general. And we have a variety of different animals, and I'll talk about one of these today. And then, of course, the most important resource we have are the MND patients. So the MND patients are always keen to contribute skin samples and blood, and that's through the Calvary Hospital. Okay, so I wanted to talk to you about what we work on, and that is we work on mice. So I've got a little video here showing you, this is a mouse which is genetically engineered with a human gene that causes motor neuron disease. And these mice replicate the human condition very well. So they're actually born normally, and they get to about 3 months of age, and if you suspend them by their tail, they have tremor in their back legs. So there's a very fine tremor. So this is a sign of motor neuron damage, and then this disease progresses over time. So over the next few weeks, this animal, you can see it's moving on a smooth surface. You can see splay in those back legs. So that's a sign of weakness. And the disease continues to progress. So here this animal is now developing some semi-paralysis in its back legs. And then we end up with a mouse like this, in which we have full paralysis in the back leg. And then we would intervene at this stage and euthanize the animal in accordance with our animal ethics. And this occurs over a 2-month period. So this is a really good mimic of the human disorder. So the question is, what do we do with these animals in the laboratory? We test drugs. We're trying to intervene and slow the progression of the disease. How do we evaluate drugs in these animals? Well, we conduct what we call behavioural tests. And this is just a snapshot of some of the tests we commonly employ in our lab. So what we have here is a video showing it's called a rotor rod, so it's a rotating rod, and it's able to measure the ability of the animal to negotiate this task, which is they're suspended on a rotating beam which gradually speeds up over time. So this is a great measure of the ability to move. The next test we have here is a treadmill test, so it's just been slowed down, so we're able to capture the paw prints of the mice over time with the disease progression. This is an example of a beam walk test. So the animal has to negotiate this beam to reach this little safe house at the end. So again, this is a good measure of coordination and balance. And then lastly, this is an apparatus we have in the laboratory which can measure the muscle strength of the animal. So we're able to place it onto a mesh and then prise it away and measure its ability to exert muscle force. So once we assess behaviour, we then would want to drill into the brain. So what we do is we conduct pathology studies. So this is an example of a mouse brain and spinal cord. And you can see it's quite small. It's about 4 centimetres in length. It's been dissected out. And what we do know is if we take the brain and we cut it in this plane, we know that the motor neurons in the brain live in this area, and they're shown up here using a purple stain. Whereas in the spinal cord, if we take a section of spinal cord and cut it cross-sectionally, the motor neurons reside in this area. And they're very clear. You can see these large purple dots corresponding to motor neurons. And we have about 20 of those on each side of the spinal cord. And what we do know in motor neuron disease is that by the time someone is diagnosed with this condition, they've probably lost 50% of those motor neurons at the time of diagnosis in the corresponding limb affected. So now what I've done here is I've turned off the lights, and what we're looking at is inflammation in the brain and spinal cord. So that's what this green signal corresponds to. So there's a lot of inflammation in MND, and what that means is that there are cells, inflammatory cells, which become aggressive and they turn against the motor neurons. So inflammation is a major problem in motor neuron disease, and we have dyes that we can go in and stain for inflammation. This is an example of what's called a Brainbow mouse. So we have the technology now to go in and we can colour individual nerve cells with every colour of the rainbow, hence the Brainbow mouse. So this is a mouse brain, and you can see it's lit up in different regions, and you can follow the different pathways and circuits. So these are the technologies that we can employ in the laboratory. My lab is very interested in molecular studies. So now we go from beyond the cell to the molecular level. And here we have our spinal cord, and we're able to then extract out and analyse individual molecules that are contributing to MND. And this is an example of a structure. It's a crystal structure of SOD1. And I told you that SOD1 was that first MND gene identified that's been affecting that Farr family for 150 years. So here is the structure, and we can look to see if there's any abnormalities in motor neuron disease. So the lab currently runs a number of projects, and these are shown here. I can't go through all of them in the interest of time, but what we're tackling is different areas of cell biology. So we're looking at metabolism of protein, energy metabolism. This is the spread of MND. This is actually a little particle that we found using a very powerful electron microscope that we think could be released from motor neurons throughout the brain that could cause the spread. we're also interested in other cells. So these are support cells. This is a T cell or an immune cell. These cells contribute actively to the disease. So the action isn't just in the motor neuron, it's in the environment of the motor neuron. So one has to take a very holistic approach in MND research. And then finally, this is what I call the business end of the lab. So this is our drug discovery pipeline, gene therapy, and stem cell therapy. So what I thought I'd do today is talk to you about this area. This is a very promising area of the lab, looking at the immune system contribution to MND. So if I come back, this is a cartoon of a motor neuron. And what we do know is motor neurons don't exist alone in the brain. They're actually surrounded by a network of supporting cells. And I've just put a diagram here showing you different types of cells neighbouring the motor neuron. And they've all got different names, so astrocytes, microglia, oligodendrocytes, schwann cells, and interneurons. And what we know is that these cells actually, over time in MND, they turn against the motor neuron. And they become what we call inflammatory, and they start to contribute to the problem. And we thought, well, is there a way in which we could introduce a cell that could address this problem? And the cell that I'm going to talk to you today about is called a T cell. So a T cell, as you may know, is the type of cell that's affected in AIDS. And it's a white blood cell, and it lives in your blood. And what we do know is that T cells can visit the brain, where they have a protective influence. So these cells are actually thought to be anti-inflammatory. So what we're trying to do is we're trying to manipulate T cells in order to restore this balance of inflammation versus anti-inflammatory response. So this project was mainly funded by the Stafford Fox Medical Research Foundation. And what we did is we developed a new approach to stimulate T cells in motor neuron disease. So how did we do this? We used a drug, and the drug's called IL-2C. And what this drug does is if you add it, if you inject it into an animal, it's able to expand T cells. So what we have now is a drug that can expand this population and really amplify up numbers of T cells. So the question is, does this drug work for motor neuron disease in the animal model? So what we did is we took this drug, so this IL-2C, and we've injected it weekly into MND mice. So the first question we had is, can it actually expand or recruit T cells? This is actually a cross-section of spinal cord in an MND mouse. And what you can see is there's these little green dots here correspond to T cells. And there's very few of them. However, if you look at the drug-treated animals, you can see that the number of T cells are increased, but they're also recruited into the vicinity of the motor neurons. So what I've done here is I've indicated T cells with arrows, and then I've shown you in red that the T cells are recruited closer to the motor neurons, which is shown in red. So this drug works in principle. It can recruit the immune system in motor neuron disease. But is it effective? So to look at this, one has to do some pathology studies in these animals treated with this drug. And I'll just draw your attention to this column here, in which we've looked at individual motor neurons. And the first response we noticed was that motor neurons appear larger. So the motor neuron, they normally shrink with disease, and this drug is able to preserve the size of motor neurons. The second aspect of this drug is that it's anti-inflammatory, and I told you at the start of this section is that we have these bad cells called astrocytes and microglia, which contribute to inflammation, and this drug is able to calm down that inflammation. So you can see a clear decrease in the green and the red signal from this tissue. So this is pointing towards a protective response of the drug. Now, did this drug benefit the animal in terms of its quality of life and survival? So again, what we did was that rotor-rod test, where we put them onto this apparatus, and we increase the speed, and we measure their ability to run across that wheel over time. And this is a curve or a graph showing you that. So this is running activity with age. And you can see that in the control group, these animals get really affected from 100 days onwards. We were able to find that this drug could provide some protection against the loss of wheel running activity towards the later stages of the disease. But what was more impressive was the effect on survival. So this is what we call a survival graph. And what we're looking at is percentage of survival over time with age. So each one of these little steps in the graph indicates that an animal has reached end-stage paralysis. And this is the typical course of the disease in these animals. So this is an untreated group, and you can see that they're dying around about 160 days of age. And this drug is able to prolong lifespan by about 2 weeks in these animals. So how do we equate that to people? So what we do know is that 2 weeks in a mouse is probably equivalent to a year and a half in an MND patient. So this is a fantastic start because we can start to play around with dose of this drug, change the frequency of injection and really boost this response. So this is a fantastic result. But what's really exciting is that we've learned that this drug has been taken forward into clinical trial as we speak. So in France and in the UK, this drug called IL-2 is now in phase 2 trial in MND patients. So this is a really encouraging step. So this is an example of going from bench to bedside. So I just wanted to finish my talk by telling you about some exciting news that we received about a month ago, and that is a major funding boost to our MND research efforts at the Florey. And this was announced from the Victorian State Government, so these are some headlines, and it also hit various newspapers and all 5 channels, primetime news. So Melbourne MND research to speed up drug tests 160-fold. So how are we going to achieve this? So what we have done is we have partnered up with several funding bodies to develop a world-first drug screening platform for MND. Because as I showed you, that previous graph with that anti-inflammatory drug, it's not the entire picture. We need probably a combination of drugs to address all the different problems in motor neuron disease. And this is a $5 million project, which has been co-funded by Victorian State Government, Fight MND, which used to be called the Cure for MND Foundation, and also the Balkan Group Proprietary Limited. And this is a very exciting venture because our objective with this platform is to really speed up the drug screening process for motor neuron disease. So we're aiming to take cells from MND patients and then to screen agents on them for therapeutic effects, and so we can then advance those quickly to clinical trial. So how are we going to do this? This is an overview of the drug screening platform. So our starting point will be MND patients. So in Victoria, there are about 400 patients. Those who wish to volunteer for the program can provide a non-invasive superficial skin biopsy. And from that skin biopsy, we can take their skin cells, turn them into stem cells, and then turn them into motor neurons. And this is an example here of the motor neurons that were on my total slide. And then what we can do is we can plate them out in these high-density assay plates to give us a testing tool. We will then take those cells and advance them towards a drug screening platform, which is shown here. So we can take many of these assay plates and put them into a machine and then treat them with drug libraries, which I'll talk about in a moment. And what we're looking for is drugs that can promote survival of motor neurons in the Petri dish. Once we identify drug candidates, we can advance them to testing in the animal models or even take them straight into patients. Because the advantage of the drug libraries that we're working with is that they are already clinically approved drugs for other conditions. So these are prescribed medications that we are seeking to repurpose for motor neuron disease. And the reason for that is obvious. We need to provide a massive shortcut to MND patients who are urgently awaiting effective treatments. So how will this work? I'll just talk about the 2 stages of this platform. So the first stage involves stem cells. And what we're doing here is we're using a Nobel Prize winning technology called iPSC. And iPSC means induced pluripotent stem cells. And the way this works is that we have MND patients who volunteer for this program. They can provide a skin biopsy. We can use technology to turn that skin cell back into a stem cell, and a stem cell can give rise to all different cell types. The cell type of interest to us is the motor neuron. We can turn this stem cell into a motor neuron. And these are actually real-life examples from our laboratory looking down the microscope at all these various steps. So you start off here with skin cells, turn them into stem cells, and then push them into motor neurons, shown here in green. And we can generate millions. This is an infinite resource of motor neurons. And what you end up here is a disease in a dish. So what it means is that, for instance, we could sample Neil Danaher and then model his form of motor neuron disease in the Petri dish, which is very important because he has the sporadic form of motor neuron disease. So we call this MND in a dish. And our prediction is that this process would take 3 to 4 months per patient. So we're talking from skin biopsy to motor neurons in a 3 to 4 month period. What then do we do with these motor neurons once they're in the Petri dish? Well, we use a technology called HDS. So HDS means high throughput drug screening, and this is a very rapid form of drug screening which was conventionally used in industry, but it's commonly being practiced in academia. And what we did was we took a lesson from cancer. So if you look at advances in cancer research, you almost hear weekly updates about some breakthrough in cancer therapy. And we thought, well, why not use the opposite approach? Instead of looking for anti-cancer drugs, why don't we look for drugs which are pro-survival, so drugs which will support the growth and survival of motor neurons. And what that means is we can tap into drug libraries. These are commercially available drug libraries which contain thousands up to millions of drugs, and then we can screen them using our platform. And our platform gives us the ability to screen 1,000 drugs a week. Now, how does this equate in terms of efficiency? We've calculated that this will increase drug screening efficiency by 160 times using this technology. So these drugs are then screened on our patient-derived motor neurons, which are in these high-density plates, and we can read multiple plates at once. So this is a massive shortcut in terms of that pathway to finding effective drugs for motor neuron disease. So that brings me to the end of my talk. So I just wish to acknowledge the lab members and our various collaborators across these different institutes, but of course our funding bodies. So I just want to draw attention to the Stafford Fox Medical Research Foundation, who were instrumental to the interleukin to the IL-2 treatment that we're developing, the Fight MND, and also the Balkan Group Proprietary Limited, who are here today, for their injection of funds into this drug screening platform. So these are very exciting times for motor neuron disease research. Thank you.